Hello students. Today we learn about the important topic microsporogenesis. It is under embryology of your syllabus. So what is microsporogenesis? The formation of microspores is known as microsporogenesis. The microspores are nothing but pollen grains. So the pollen grains are present inside the anther lobe. So we learn about the structure of stamen also because it is the male reproductive organ of the flower. So we learn about the stamen structure. So the stamen has a stalk of the anther. So this is the filament. It is the basal part of the stamen. So it is the sterile part. The upper part of the stamen is known as anther. So it consists of, in majority cases, two anther lobes. Such anthers are known as dithecous anthers. In some cases, the anther consists of only single chamber, that is known as monothecous. So in dithecous anthers or monothecous anthers, the sporangia are present. So in dithecous anthers, in transverse section, we observe four locules. So it is known as tetrasporangiate anther and dithecous anther. In monothecous anthers, only two chambers are present, that is bisporangiate anthers. So the anther is dithecous, consists of two lobes and four chambers, that is tetrasporangiate type of anther, like this. So how these microspores are formed inside the anther lobe? So before we start with the structure of anther, the anther is formed like this. Starting, there is a single epidermis, outer layer. It consists of a mound of tissue. So, initially, it is oval shape. Gradually, it is differentiated like this. The tetralocular shape is arranged. In the starting structure, it consists of outer epidermis and a mass of undifferentiated tissue. In later case, below the epidermis, there are some cells differentiated. Those cells are known as archisporial cells. These are fertile cells. These are hypodermal in nature. That means below the epidermis. These archisporial cells, they may be group of vertical rows of cells. In TS, it shows like this, a plate-like structure. These are known as archisporial cells. In some cases, only single vertical row of cells are present. In TS, it is only single cell is differentiated. And this archisporial cell or cells, they are periclinally divided and consist of or form parietal cells on the upper side and sporogenous cells on the lower side. These parietal cells, which are formed outside the archisporial cells, by number of periclinal or anticlinal divisions, they form anther wall. And the sporogenous cells, which are formed below the archisporial cells, they again undergo number of mitotic divisions and differentiated into sporogenous cells. These sporogenous cells, later they undergo meiosis and form four tetrads. But these parietal cells, which are formed outer region, by number of periclinal and anticlinal divisions, they form anther wall. The anther wall consists of outermost epidermis, and next to the epidermis, it consists of endothelium layer. This endothelium layer consists of radially elongated cells and consists of fibrous thickenings radially and tangentially. This endothelium cell layer helps in anther dehiscence, which is hygroscopic in nature. This layer of cells consists of fibrous thickenings, but these thickenings are absent at the region of stomium, and this endothelium layer, by contraction and expansion uh, function or mechanism, they, it helps in dehiscence of the anther and liberates the pollen grains outside. And this endothelium lacks the fibrous thickening in cleistogamous flowers, that means unopened flowers. Next to the endothelium, the parietal cells forms middle layers. The middle layers are present 2 to 3 layers in number, and these layers are ephemeral in nature. That means they intact only in early stages of the anther. That means pollen grains development. That means up to the meiosis or up to the pollen mother cells meiosis stage. Later, these middle layer cells perish or disintegrated. Sometimes these middle layer cells also store food materials and gives nourishment to the developing pollen grains. And next to the middle layers, there is a single layer of tapetum. It is completely surrounds the locule of the anther. So this tapetum is either in single layer or in multiple layers. In majority of the flowers, it is in the form of a single layer. This layer consists of cells which are somewhat elongated cells and parallel. It is like this single layer, and consists of prominent nucleus and dense cytoplasm, and this layer of cells multinucleated in majority of the cases, and in some cases it is single nucleated. Example, in multinucleated condition, GMA. So this layer, it completely surrounds the anther locule, tapetum. It is also ephemeral in nature. It is remains as is at the stage of first meiotic stage of the pollen mother cell meiosis. The tapetum function is it gives nourishment to the developing pollen grains, and also it forms the wall layer of the pollen grains. The tapetum is two types, that is amoeboid tapetum, and another one is it is glandular tapetum or secretory tapetum. Amoeboid tapetum means it is present. It is a primitive type of tapetum. In this, the tapetal layer cells break and form and releases amoeboidal masses, that is protoplast masses, into the locule, and that protoplast masses fuses and form a layer around the pollen grains, and they form or they gives nourishment to the developing pollen grains, and also sometimes help in exile wall formation. And another type is secretory type of tapetum. This tapetum, it secretes some pro-ubish bodies. They consist of some carotenoid esters and carotene, and later they transferred into ubish bodies. Those bodies also help in the formation of pollen grain wall. And this this is the secretory tapetum or amoeboid tapetum. So there are two types of tapetum layers in case of angiospermic plants. Next to the tapetum, there is a sporogenous tissue. It is formed from archisporial cells. The sporogenous tissue remain directly, they divide and form spore mother cells, or they undergo one or two divisions of mitotic divisions and form spore mother cells or pollen mother cells or microspore mother cells. These microspore mother cells later undergoes meiosis, and they give rise to microspores, which are at first in the form of tetrads like this. Later, they released from the tetrad and forms like this. They release are independent or separate from the tetrad, and like they form independent pollen grains. These are round in shape. This pollen mother cell, it undergoes meiosis. Again, it is two types of divisions. That means the successive type and simultaneous type. In this chart, it is a simultaneous type of division is given. It example Nicotiana and Magnolia. The pollen mother cell undergoes first meiosis and later second meiosis, and the wall is formed at the end of second meiosis uh, and gives pollen tetrads. So the four pollen grains initially attached like this, and later they separated from this tetrad and released like this, separated like this. Later these pollen grains present in the locule, they released or liberated out from the stomium. That means by the shrinkage of endothecium layer. Next, the pollen tetrads are the arrangement of the pollen grains or microspores in the tetrad. In four types of arrangements are present. That is, they may be in tetrads like this, or they are arranged isobilaterally like this, or they are present in decussate manner. So, in the basal region, two spores are present. In the upper region, opposite to this two spores, again two spores are present. This is the isobilateral. At right angles to the lower spores, upper spores are present. That is the decussate type. And in some cases, T-shaped arrangement is present. Like these two spores present like this, and the upper two spores like this. In some cases, four spores are arranged like this, linear type. So depend of the species, there are different types of arrangements are present. In some cases, as Asclepidaceae, all the four types of arrangements present. So these spores released outside of the anther at the time of dehiscence. At the time of dehiscence, the anther lobes dehisce like this longitudinally, or they, in case of monothecous anthers, they dehisce transversely. In some cases, the anthers dehisce like this, valvular dehiscence, like this, or in some cases, the anther dehisce like this. At the tip of the anther, two pores are present, and through these pores, the anthers, the pollen grains are dehisced outside. So this is the incasia species like that. So this is the dehiscence of anther. So after coming outside of the pollen anther, these pollen grains they developed into like this. They form the male gametes, and later stage is known as male gametophyte development. So this is the formation of pollen grains in anther locule. Thank you.